you probably heard before in other classes that if you take a red blood cell and put it in an isotonic solution, it'll maintain its normal morphology and shape, morphology shape. If you were to take a red blood cell and put it in a hypotonic solution where there's more solute inside the red blood cell than inside the solution, what's going to happen is that water is going to flow into the red blood cell, into those aquaporins, and basically it's going to swell and enlarge, and eventually it can break open. It's called lysis. It can break open and the red blood cell will be dead. The opposite happens if you take a red blood cell and put it in a hypertonic solution. So basically, if there's more solute on the outside of the red blood cell than inside of it, the water is going to leave the red blood cell. And we call this crenation because it kind of looks like a shell. And another thing is, you could just call it more simply shrivel. But crenation is the scientific term. Shriveling is just another descriptive term. And in the body, you can imagine that it's really critical that our cells are isotonic with the blood and tissues so they can maintain their normal morphology. So in terms of facilitated diffusion, I want to talk a little bit more about this. I had a diagram about facilitated diffusion a few minutes ago, and I just want to show you that glucose is an example of a molecule that can move with its concentration gradient, using facilitated diffusion and carrier proteins. So if we have more glucose outside the cell and less glucose inside the cell, glucose molecules can interact with the carrier protein. The carrier protein will basically detect the glucose and then, close on this side and open on the other side, letting glucose passively enter the cell, following its concentration gradient. This does not require ATP. It is just diffusion mediated. So that was passive. Now I want to show you an active transport pump. And this is one of the most important active transport pumps in the body. This is the sodium-potassium pump. And what it does is it uses the energy from ATP hydrolysis, going from ATP to ADP plus phosphate, to pump out sodium, specifically 3 sodium, from the inside the cell. It pumps it outside the cell. And at the same time it pumps sodium out, it'll bring in 2 potassium. So we're saying 3 sodium out, 2 potassium in. And critically, both of these molecules are being transported against their concentration gradient. That means that there is more sodium outside the cell already than inside the cell, but yet we're still pumping more out with the sodium. And there's more potassium on the inside of the cell than on the outside. However, we're still pumping that potassium in because there's more potassium on the inside, less potassium on the outside. We're still pumping that in the cell, right? And what this does is it helps to, one, maintain a difference in the resting membrane potential from the inside the cell to the outside the cell. So because we're pumping out 3 sodium, we're bringing 2 potassium in. This helps to maintain a resting membrane potential of negative 70 millivolts. And that's really important for our nervous system to send impulses throughout the body. And another critical thing that this process does is to allow the cell to use the energy in the sodium and potassium concentration gradients to do work. And we're going to take a look at an example of that in a minute. This is also critical because it also helps to maintain osmolality. Basically, we want to have everything be isotonic in the body. We want to have the osmolality of a cell be the same as the osmolality of the blood and tissues. So the sodium-potassium pumps helps cells to maintain an isotonic relationship between the inside of the cell and the outside of the cell. So those are three critical things that these sodium-potassium pumps do, and cells spend a lot of energy, they use a lot of ATP to maintain this process of having this concentration gradient maintained, where you're having more sodium on the outside of the cells and more potassium on the inside. And this is a critical thing to remember: more sodium on the outside, more potassium on the inside of cells. And this is created and maintained by the sodium potassium pumps that are always on in the cells of our body. So next I want to show you an example of how the sodium concentration gradient can be used to bring in glucose against its concentration gradient. So I want to start with the second frame right here, showing you that sodium concentration is higher on the outside of the cell. In this cell, glucose concentration is higher on the inside. So what can happen is that the sodium and glucose can bind to the co-transport channel. When they're both binding, the energy of the sodium concentration gradient will allow for glucose to be brought in against its concentration gradient. So again, the energy of the sodium concentration gradient is now moving glucose against its concentration gradient to the inside of the cell. And then both the glucose and sodium will be inside the cell now, right? So basically the sodium-potassium pump stored what created this energy in the concentration gradient for sodium. And now that concentration gradient is doing work by now bringing in glucose against the concentration gradient. So a very important example of how important the sodium-potassium work is. And there's going to be way more examples coming as we talk more this semester. So I love this next slide because it shows a big picture of how these different pieces of the puzzle that I've been talking about fit together in a cell in our body. So let me explain what we're looking at, because again, I'm excited about this figure. So here is an epithelial cell, and then below the epithelial cell we have a capillary with blood. And then above the epithelial cell we have the lumen of the kidney tubule, or the small intestine. So I'm going to talk right now more focusing on the small intestine because I think it's easier conceptually at this point than the kidney tubule. But this process occurs in both of those tissues in the body. So we call this membrane with these microvilli an apical membrane, and then the membrane touching the capillary is called the basolateral surface. So what I want to first highlight here is in the basolateral surface of the epithelial cell, we have a sodium-potassium pump, right? So it's using the energy of ATP hydrolysis to push out sodium against its concentration gradient. It's using that same energy to bring in potassium against its concentration gradient, right? Making these sodium-potassium concentration gradients. So once that is established, right, we have low levels of sodium in the cytoplasm, and we tend to eat a diet that's high in salt. So the intestinal lumen tends to have high levels of salt in it, and if you eat carbs, you will have glucose inside the intestinal lumen too. So what happens is the co-transport transmembrane protein is going to be found in the apical membrane of this epithelial cell, and the concentration gradient of sodium is going to provide the energy needed to bring in glucose against its concentration gradient. So we know sodium's higher on the intestinal lumen side, lower in the cell. diffusion is going to want to bring that sodium from the intestinal lumen to inside of the cytoplasm. That glucose is not going to want to be doing that, right? It's not going to want to do that because it's in a low concentration on the intestinal lumen as compared to this epithelial cell. The glucose is going to want to be going out of the cell and into the intestinal lumen. So to get the glucose in, our intestinal epithelial cells are going to use the energy provided in the sodium concentration gradient to bring in glucose into the cell. That glucose, even though it's going against this concentration gradient, it will be brought into the cell via cotransport by the sodium glucose cotransporter. So remember, when we're eating a meal, we need to get the glucose molecules from our diet to be circulated throughout our body, right? So yeah, we have all this glucose in our intestinal epithelial cell, but how does it get into the blood to be circulated to tissues and cells in our body that need that energy? So the glucose concentration is higher in the cytoplasm of the epithelial cell and lower in the blood. So now a glucose facilitated diffusion transporter can just allow for the concentration gradient of glucose to be allowing glucose to be having facilitated diffusion, moving without needing ATP from the cytoplasm of the epithelial cell into the bloodstream, and then from there it can be taken up by cells that need that energy. So I just want to remind you about the processes of endocytosis and exocytosis. I know we talked about these before. Both of these processes require ATP. So for endocytosis, we have these molecules on the outside of the cell that the cell wants to take in. It'll invaginate the plasma membrane, form this little pouch, and then the pouch will be pinched off to create a vesicle. Exocytosis is the reverse process, right? And we know that the exocytosis vesicles are going to be leaving the Golgi, right? And these vesicles full of whatever is destined for secretion will be going up to the membrane. It'll fuse with the membrane and then release the product for secretion.